Welcome back to sessions in molecular biology. We will be continuing with the non-coding RNAs. The non-coding RNAs, which we have already discussed in the one of previous presentations, include the microRNAs and the small interfering RNAs. In this presentation, we'll be looking into the piRNAs. Okay, so we'll just summarize what happened with the microRNAs and small interfering RNAs. MicroRNAs and small interfering RNAs, they are small stretch of RNAs, double-stranded RNAs with 19 to 24 base pair in length, and they are usually encoded at widespread locations. And in humans, more than 1424 microRNAs are found, almost like around 1500. And the target sequences include the mRNAs and many others, and by which they bring about the arresting of the transcription, post-transcription gene silencing. Okay, examples are microRNA 15, 6, or 16, microRNA 124A, etc. They include evolutionary conserved sequences, and they are endogenously produced single-stranded small non-coding RNAs. They are derived from endogenous short hairpin transcripts. Coming to the siRNAs, which we have already seen, they are partially complementary passenger and guide RNA strands. I hope you remember what is guide RNA strand. That is when after the separation of the the strands of the double-stranded RNA, one of them it gets what you call degraded, and one it remains complexed with the Argonaut family. That one, the one which remains, it is the guide RNA, and the other one is the passenger RNA. Okay, the RNA which is found in the pre-risk is what is referred as the passenger RNA. Now, it is involved in post-transcriptional gene silencing, and it is mediated through the risk-mediated degradation of mRNA. Okay, so these are the major concepts which we have discussed in previous presentations with regard to the non-coding RNAs. In this presentation, we'll be looking into piRNAs. piRNAs, the size ranges from 26 to 31 base pairs, and they're usually found in clusters grouped together within the intragenic regions. And they actually help in repression of transposons and also methylation of DNA. They include the largest group of the non-coding RNAs, and unlike the miRNA and siRNA, they include the single-stranded non-coding RNAs. They are generated by the dicer-independent mechanism, because as we have already mentioned, in the siRNA and the microRNA, these are produced by the involvement of dicer molecules, but here it is dicer-independent mechanism for the piRNA synthesis. Okay, now examples include the piRNA which targets the Ras GRF1, line 1, and IAP elements. Okay, we'll see in detail what is piRNA. piRNA, it is simply the PIWI interacting RNAs. This is a protein actually, and it has been shortened to form the piRNA. These piRNAs, they are small endogenously produced RNA molecules which ranges from 21 to 31 base pairs in length. Nucleotides in length, and these actually form a part of the genome's natural defense, especially against transposons. Specifically against transposons, and they may be involved in post-transcriptional gene silencing and other epigenetic functions. Okay, these are short double-stranded sequences are here in the formation during the formation of a synthesis of piRNA. Short double-stranded sequences are processed by dicer-independent mechanism, and the resultant single-stranded RNA, it gets associated with Piv proteins to produce the piRNA. Okay, now they participate not only in the post-transcriptional gene silencing but also transcriptional silencing as well. So how it happens, we can see. The piRNAs, they are the most abundant, as we have already mentioned. It is the largest group of non-coding RNAs, and they have been best studied and characterized in Drosophila melanogaster. Examples include the MILI, MIVI, MIVI2 in mammals, And these small RNAs, they do not arise from hairpins, unlike what happens in the case of what you call the short hairpin RNA, the micro RNA, or the siRNA. Here in piRNA, during the biosynthesis, we cannot see any formation of hairpins. Okay. Now, usually it is the germ cells of the animals that express this particular piRNA, and the piRNA in the germ cells, they are responsible for suppressing the movement of the transposons in the germline. Okay, the piRNAs, as already we have seen, it get their name from the proteins with which they associate, and these proteins are known as the PIWI, and the PIWI belongs to a subclass of Argonaute family. I hope you remember the Argonaute. It is the Argonaute protein which is associated with the risk complex, the formation of the risk complex. Okay, which is involved in the formation of siRNA, which is involved in the RNA silencing by the siRNA and the microRNA. Okay, the roles of the PIWI proteins, it has been best studied in Drosophila, that is the fruit flies, where deletion of the proteins leads to the defects in the suppression of the transposon movement in germ cells, and ultimately the failure to form the gametes. Okay, the piRNAs and their associated PIWI proteins, they are required for the successful gamete formation in the male mice. So in the Drosophila as well as in mice, the role of piRNAs have been well studied. Now, what are the how it is being actually produced? Okay, it is by way of one mechanism for the synthesis of piRNAs, ping pong biogenesis pathway. We can see with this figure. Okay, here you can see what is happening. Now, the piRNA, unlike the what you call the siRNA and microRNA, here the piRNA synthesis, it depends upon the single-stranded RNA endonucleases, which is referred as zucchini. Okay, and this zucchini, it acts on long single-stranded the RNA transcript. Here you can see the precursor piRNA is being produced. Precursor piRNA transcript is produced, and this enters the what you call cytoplasm, where it undergoes the primary processing. And during the primary processing, this length of the primary precursor piRNA, or like precursor piRNA, the length is shortened by way of what you call the cleaving. Right, and it is reduced in length, it is being shortened. That is referred as the primary processing. Later it undergoes the secondary processing, and the secondary processing is by way of ping pong amplification. Okay, during this process, what happens is these mRNA, the precursor what you call piRNA, it gets loaded onto the PIWI proteins. Here you can see it is getting loaded onto the PIWI proteins. Okay, and this is the final step for the involvement of these PIWI, what you call the piRNA getting loaded onto the PIWI protein. This complex, it gets involved in the post-transcriptional silencing, and that is the first kind of the what you call the RNA interference, and this is brought about by the degradation of the transposon RNA. The RNA which is produced from the transposons, it is being silenced by the PIWI protein loaded piRNA molecules. Okay, this complex, it brings about post-transcription silencing. Now, the second kind of mechanism is the transcription silencing. What happens is this PIWI protein loaded piRNA, it can bring about DNA methylation of the transposons or even repetitive elements on the DNA. And so this brings about the transcriptional silencing. It is mediated by RNA directed DNA methylation, and this is brought about by specific kind of piRNAs, for example the MIV2, etc. Okay, so here it can bring about post-transcriptional silencing as well as the transcription silencing. Okay, what are the functions? It can bring about degradation of spermiogenic mRNAs, degradation of maternal mRNAs, production of genic piRNAs, and it also gets involved in acrosome formation, germ cell development, cancer cell migration and metastasis. So these are actually different roles played by piRNAs in different animal species. Now, what are the differences between the piRNAs and the SI or the microRNAs? piRNAs are longer than the other two RNAs. The microRNAs, they act only in post-transcriptional gene silencing, while the piRNAs, they can bring about both post-translation as well as transcriptional silencing. Okay, the transcriptional gene silencing is what is referred as the epigenetic silencing, and the piRNAs, they are subjected to amplification, ping pong amplification, and microRNAs are dicer dependent, but piRNAs are independent of dicer. Okay, so